Hi, my name is Kathy Seiler. I have a PhD in cell and molecular biology, and I'm the manager of a biobank here at a hospital in Phoenix. My mission as a biobank manager is to make sure that researchers have the resources that they need to better understand, diagnose, and treat diseases. So in this talk, what I want to do is describe to you what a biobank is, how it is that we biobank, and then give you one example of how what it is that we do is able to impact research and also patients' health and well-being. So to start off, what is a biobank? You might be thinking about a regular bank which holds currency. So does a biobank, but this currency isn't money, it's actually some type of biological specimen. Now this could be anything from seeds to animals to plants, but for the sake of this talk, I'm going to focus exclusively on human biospecimens. So this is anything from hair, nails, blood, urine, toenails, really whatever you collect from an actual person. Now, like a bank, we actually keep these samples securely held, in this case at very, very cold temperatures in liquid nitrogen freezers that are at minus 175 degrees Celsius. And that keeps these samples safe so that they'll be really useful for researchers when they're ready to study them. Similarly to banks, this currency is used to buy something, but instead of buying things, it actually buys knowledge. When we're able to distribute these specimens to researchers, they're able to use them in a variety of different ways to better understand the characteristics of these samples. Now, let's use cancer as an example. If you want to understand cancer, you want to understand cancer cells. And one of the best sources of cancer cells would be the actual tumor tissue cells themselves. And in order to access those, you might reach out to a biobank and see if they have examples of tumor tissue cells available within their biobank. So in the case of my biobank, we very often have cancer tissue available because what it is that we do is we work directly with cancer patients. We help them understand what it is that we're planning on doing in a process that we call informed consent. We make sure that they understand how we're going to collect their tissue, how we're going to store it, and what we're going to do with it. And it's only with their permission that we store and we use these samples. So once we talk to a patient and they say they agree to be a part of the biobank, we then, in either the OR or within pathology, if there's any tissue sample that's left over that is not needed for diagnosis, that would otherwise be thrown away, this is something that we store in a tiny little tube inside one of those liquid nitrogen freezers waiting for a researcher. And again, we never interrupt diagnosis. Diagnosis and patient care is the most important part. So if there isn't enough tissue available after diagnosis, that's okay. We just let it be. So at that point, a researcher is able to reach out to me and say, do you have any samples of this particular type of cancer or disease that I'd be able to study? And again, in a very ethical manner, we are able to provide these samples to researchers. Now let's talk a little bit more about how it is that researchers use these samples. So I think an interesting example is the case of breast cancer. A very good friend of mine was actually diagnosed with breast cancer in graduate school. And so I got to see a lot of her journey going through breast cancer diagnosis and treatment. Now in her case, when she had her lumpectomy, they looked at the cells within that particular piece of tumor, and they found out that there was a mutation in this particular gene called HER2. This HER2 mutation made it so that it was specifically targeted by a drug called Herceptin. In this case, the test looking at the biospecimen was actually developed by looking at lots of different breast tumor samples and comparing those breast tumor samples to samples from patients who didn't have breast cancer. Then these differences could be used to identify things that were changed. In this case, this gene Herceptin. And in this case, this gene that was changed, they developed a drug to it that specifically said, instead of making the cells grow like cancer cells like to do, let's make these cells stop growing. And so in her case, she was able to take Herceptin, and I'm happy to say that she's doing very well in her full remission. So I hope in this brief talk I was able to give you a little bit of insight into what it is that a biobank does. We store biospecimens to be used for researchers. I hope I also gave you a bit of an idea of how it is that we actually store these specimens. We work directly with the patient, we make sure they're fully informed, and then we store these samples within our biobank. And then I hope you understand our example of how it is that these can actually be used to better understand, diagnose, and treat cancer. Maybe the next time that you get approached by someone to donate a biospecimen of your own, you may actually say, I understand this and I'd be happy to participate. Thank you so much.